Okay, good morning, good afternoon, depending on where you are. Welcome back to the NIMH Center meeting. It's my pleasure. My name is Amanda Brown. I'm a co-director of the Developmental Core, and it's my absolute pleasure today to introduce our speaker, Dr. Yoelvis Garcia Mesa. I've known Dr. Mesa since 2012. He did his bachelor's degree in microbiology at the University of Havana, Cuba, and then his master's in the neurosciences at the University of Barcelona in Catalonia, and stayed there at the University of Catalonia to complete his PhD in biomedicine in 22. He's currently working as a senior research associate in Dr. Jonathan Carnes' lab. And he moved, we were speaking at before we started, moved from the Alzheimer's field to the neuro HIV field in Dr. Carnes' lab back in 2012. And we are just excited to know about his cutting edge work producing microglial cell lines that physiologically represent the microglia that we so robustly study and need to understand in the neuro HIV field. In 2016, he was in the Department of Neurology at the Perelman School of Medicine at the University of Pennsylvania, where he worked with Dennis Colson for more than 5 years. And there, he was really then understanding the importance of early injury to the brain, focusing on the role of heme oxygenase 2 in the development of SIV and HIV neuropathogenesis. So we're really excited to have you here today. You can see the title of his talk there. And so, Elvis, please take it away.
Okay, thank you, Amanda, for the nice introduction. So, also I want to thank the organizing of the seminar series for giving me the opportunity to be here today presenting my work. As you can see, I will be talking about adenosine and how the adenosine can be having a role in protecting from HIV neuropathogenesis. So more likely we all are here today because we are very much interested in understanding how HIV can be affecting the brain and inducing neuropathogenesis. As we all know, the brain dysfunction in HIV infection can evolve despite the viral suppression. So it has been accepted that maybe around uh, 50% of the people with HIV can be suffering from HIV-associated neurocognitive disorder. Uh, Therefore, if there is around 1 million people infected with HIV in United States, around 500,000 people with HIV then may be suffering from HAND. So, but the question here is what factors can be contributing to the development of the um, HIV-associated neurocognitive disorder. So there are some group or there are many people that believe that the antiretroviral therapy probably is not strong enough to silence an HIV in the brain, and there will be this low level of viral replication, and then this can be contributing to the disease. And there is another group or another people that they are working with immune activation. They believe that probably the immune activation that is kind of chronic in the HIV people, it could be the reason for the development of the hands. There is another group of people that they are working with antiretroviral. They know that the antiretroviral is very important in silencing HIV, but they also believe that, or they have been showing how the antiretroviral can have some toxicity in the brain and then also can be a contributor. So as Amanda said before, I was also working trying to understand how the early HIV infection can be contributing to the development of the disease. I believe that probably those early events that are happening as soon as the virus get into the brain, they can be responsible for how the brain will be responding later on, how the disease will be developed or not. So I'm talking a little bit about the course of the infection. As you can see here in this graph from Dr. Colson, very early after the infection, even before of the seroconversion, you will have a meningitis. So then probably around the seroconversion time, you can have some kind of inflammatory syndrome in the brain. So we were at Dr. Corson Lab. I was trying to model those early events, and here you can see in the macaque using SIV. Uh, 5 day post-infection, we were able to detect a plasma viral load. However, a 5 day post-infection here in the bottom, we didn't see any viral load in the brain. However, we saw astrogliosis already going on in the brain, even we were not able to detect um, virus in the brain. So meaning that probably, the inflammatory process in the brain are starting as soon as the virus get into the periphery. So continuing with this graph, we know that after the establishment of the antiretroviral therapy, hopefully the antiretroviral therapy is successful. So the virus is gonna go down and it's gonna start being undetectable. The CD4 positive T cell will start going up. However, they never gonna go back to the level that they were before. And we will see this kind of inflammation, immune activation, and oxidative stress that will be, somehow like chronic happening in the HIV patients. So in our group, we have been working with the hypothesis that probably this neuroinflammation can lead to cycle of HIV reactivation of latent virus in the brain, and these may be contributing together with other factors to the development of the disease. So we were very much interested in them, how these can be affecting the crosstalk mechanism that are happening in the brain between microglia, astrocytes, and neurons, and specifically how HIV can be breaking this crosstalk that is very much important for keeping the homeostasis in the brain. So how the HIV is affecting this and how HIV can be breaking this system and how the neurons and astrocytes are contributing to recover the homeostasis and silencing HIV. So many years ago here in this laboratory, when I was here before, with Dr. Alvarez Carbonate, we start putting together, where first we create the H69 cell line. I'm gonna talk a little more about that cell line later on. But we create the cell line. That cell line is infected with a single round HIV and is carrying the GFP as a way to identify the cells that are expressing HIV. Uh, we activate those cells and we put it together with healthy neurons. And we saw that when we keep higher radio, like more neurons and less microglia, so the neuron were able to silence an HIV. So, however, when the neuron were exposed to some kind of insults like methamphetamine, 100 nanomolar, this concentration that I already talked in the ISMV meeting, this concentration is not doing anything over the HIV in the microglia, it's not activating the virus at all. However, 100 nanomolar is able to do some damage to the neurons. We then were able to see that the virus was getting reactivated. And very similar with poly-IC and the combination of those two drugs. So meaning that probably, the main summary from this work, it was that when the neurons are healthy, they will be releasing some kind of factor like glucocorticoids, fractalkine, and that will be keeping the microglia in homeostasis, and that will be keeping also the HIV silence. However, the neuron is exposed to some kind of insults like methamphetamine or another inflammatory cytokines, then they will have some damage, and that will be releasing DAMPs molecule that will be activating the microglia, will be breaking the homeostasis, and will be reactivating the HIV. Okay, but then what happened with the astrocytes? So till that point, we were working with the neurons, so I was very much interested to know what are the astrocytes doing in this crosstalk mechanism. So we know the astrocytes are around 50% of the cell in the brain. We know they are very much involved in homeostasis. They are able to promote neuronal survival. We know also that when the astrocytes are activated, they can be contributing to inflammation very much, but they also can be contributing to the resolution. So the astrocytes also have been involved in many neuropathogenesis like Alzheimer and Huntington. So with this on my hand, I start differentiating astrocyte from iPS. I am using this protocol. It's a long protocol. It takes probably around 4 months to have a 95% pure astrocyte that they are mature and express the astrocyte marker. This protocol is in collaboration with people from, specifically Dr. Wilber Romero Fernandez from Vanderbilt University in Nashville. In this protocol, we only use small molecules. We use growth factor, but we don't use at all FBS. We don't use fetal bovine or any other kind of serum. This is very important to keep the astrocyte very far from being activated. So that's also why this protocol is taking a long time, because as other people have been showing, having a little bit of FBS in the media can accelerate the differentiation, but we are doing this without FBS at all. So basically, just for you to see at day 60 of differentiation, you will have somehow a good amount of astrocyte, a good amount of neurons, and you will still have a lot of MPC. You can see here all these blue nucleus that they are not expressing MAP2, neither GFAP. However, as I said before, around 4 months later, you will have probably 95% pure culture. Basically, you don't have more MAP2. You still have probably some MPC here. You see this cell here that is not expressing GFAP. This other here that is not expressing GFAP. But I can tell you all of them are expressing Vimentin. So at this point, what I did, it was, take my H69. This, as I said before, this is a cell line that I create by immortalizing primary human microglia that we got from science cells. Those cell ones, they were immortalized. I infected with this single round HIV virus that was carrying the GFP as a way to see the activation of the virus, the presence of the virus. We then selected by a clone that they were in latency, and this is the case of the H69. Most of the time, the cell can be in latency if you keep it with the right condition and the right media. They respond very easy to inflammatory stimulus. You can see here how they respond to TNF alpha. So we took that line, we activated with TNF alpha, 400 picogram of the TNF alpha, and then we add the cell trace violet to be able to identify the cells in the co-culture, and then we started putting the astrocyte, the microglia with the astrocytes. So here on the left panel, you have only microglia alone, 24 hours after the stimulation, they already are going back to latency. They were like 40%, where when we added the astrocyte to the astrocyte, the microglia in a radio 3 astrocyte, 1 microglia, or 15 astrocyte, 1 microglia, the astrocyte were silencing HIV in those cells. Yes, of course, we have a control in a meaning that our microglia was the same number that we have with astrocyte microglia together. Sorry. So we repeat this experiment a few times, but we were concerned because the microglia, they divide fast compared to the astrocytes. It's a line, So the other thing here, we were worried that they were losing the cell trace. We were also worried the astrocytes were somehow phagocyting, rest of the microglia that they were having this, and we were misunderstanding the situation. So what I did is we took this lentivirus overexpressing lentivirus carrying the RFP, and we overexpress the RFP in the H69. So that way we selected for the red fluorescent protein, and we have a pure 100% red cells outside of the GFP. And then we put together this microglia again with astrocytes. And in this case here, I'm showing you the 7-1 radio, 7 astrocyte, 1 microglia. As you can see here, after the cells were stimulated, 80% of GFP, HIV expression, went down to 50% in just 48 hours of co-culture. So one of the things that we were worried, is this reversible? Because if this is happening in the brain, it needs to be reversible, Other way, it was just noise. So we were concerned, and we treat our co-culture with TNF alpha, just inflammatory cytokine, that we had shown many times is able to reactivate the virus from latency. And again, we saw how very nicely the TNF alpha 10 nanogram per milliliter was able to reactivate the microglia that was in latency in the co-culture with astrocytes. So at this point, I start thinking, okay, what is going on? How astrocytes are modulating HIV in the microglia cells. So this is a paper that we published many years ago, where we are explaining how this modulation can be happening between microglia, astrocyte, and neurons. I start reading what can be released from astrocyte that can be anti-inflammatory. Uh, so some people were proposing interleukin 10. I tried interleukin 10. I didn't see anything that interleukin 10 was reducing HIV expression in the microglia. I tried different growth factors like GDNF or BDNF, and I didn't see really any significant latency happening after I treated microglia. But then I realized that I was taking this in the wrong way, you know? So if we have microglia that is infected, we have astrocytes, somehow we are modeling the hen brain. So we have the microglia that is activated. So probably there is a lot of DEMP molecule there, like ATP and ADP. So, okay, the microglia are not responding to growth factor from astrocyte, like the typical healthy brain. So let me see at least if the microglia are responding to this activator, because maybe the microglia are not responding. So then what I saw, for my surprise, it was the ATP and ADP in high concentration, 5 micromolar, somehow high concentration, 10 micromolar, was taking the H69 from like 20% to 10 to 5 to 4 and to 3. So somehow the ATP, they were an ADP that they are somehow activated were inducing latency in microglia. So I got a little confused. I went out there to read a little more. And then I realized about this paper, and I realized people have been working with adenosine for some time already. Saito-san. Maybe in 1991, it was the first paper showing that adenosine can have a role in regulating inflammation in the HIV. So they were proposing that when the cells are activated, they release a lot of ATP. The way of this ATP can self-regulate the system is when the concentration go up, then the ATP can be catabolized to adenosine. However, in the rich adenosine environment, you will have less T cell activation, you will have less migration, less cytokine production, and at the end of the day, less cytotoxicity. So they were even proposing different pathway, different target that you can even modify to take the balance one way or other. So we were then very much interested in this one here because we were using ATP and ADP, and we were thinking maybe the ATP and the ADP is being catabolized with the CD73 is a ectonucleotidase. So we were very interested in this step here. And what we did then, it was just treat the cells 2 hours before with 2 micromolar of the CD73 inhibitor. And whatever we saw, it was just then the ATP and the ADP were not inducing any latency, meaning suggesting that it was really adenosine and not the ATP or the ADP which were or which was really inducing or responsible for this latency happening in the microglia. But then what happened in the co-culture, So if the adenosine is responsible for that, what I did was I took different antagonists for the different receptors, and I put it together. I did a mix of 1 micromolar of those antagonists, and I put in the co-culture, and indeed the latency was stopping from happening, probably more than 90%. And we saw, that maybe, or was suggesting that maybe, yes, adenosine was somehow responsible for this astrocyte inducing latency in the microglia. From where is coming this adenosine? We at this point didn't know, but we believe that probably is just that the activation is increasing the concentration of ATP, and then this ATP is being catabolized to adenosine. So, early before I added the ATP to the microglia, so if they don't express CD73, they shouldn't be having this effect. So then I started staining for CD73, and here you can see the H69 are expressing CD73. The astrocytes also are expressing CD73. But what is more interesting, when we put together the astrocyte and the microglia, we saw that the concentration of CD73 was really increased, meaning that probably the astrocyte or the microglia and astrocyte together were inducing somehow overexpressing the CD73, and probably that's how astrocytes were modulating the HIV in the microglia. So I went there out and see that there were already some people already talking about how in this experimental autoimmune encephalomyelitis model, they were showing that when the T cells are coming activated, they get in touch in this showing here in this coronal section. When they get in touch with astrocyte, they will be overexpressing the CD73. Astrocyte will be inducing overexpression CD73, and that is one of the mechanisms that they can be controlling T cell activation. They were even proposing that probably is one of the mechanisms could be astrocyte releasing TGF beta. So here, we have 4 receptors for adenosine: the A1, A2A, the A2B, the A3. Here in this graph, this is one of the reasons the adenosine is called the guardian of the brain. There is different modulation for different receptors. One of them can be activated while the other can be inhibited at the same time. In the cerebral ischemic injury model, in the acute phase, you will have activation of receptor A1 and the receptor A3. That will be keeping the synaptic transmission down, will be keeping down the energy consumption, the neuronal activity, and somehow the nerve inflammation. Also, that will be helped by inhibition of the A2A and the A2B. However, later on during the recovery, it has been shown that the A2A activation is being good, and the inhibition of the A1 and A3 to start recovering the neuronal activity and the synaptic transmission. So with this in hand, we went there to see really what adenosine was doing, the agonists were doing in the receptor. So we went there, we got adenosine receptor agonist for the A1, and the antagonist, we saw that the agonists were inducing latency compared here to in gray the untreated and the DMSO control. And we saw that the agonist for A1 was somehow inducing latency, the same, a little better with the A2A, rigadenosone. It was a little more potent. The A2B was not that nice. Also, the A3. So the A3 here, the NICA, is a very strong agonist for, it have a lot of affinity or a strong affinity for the A3, however, in high concentration can be binding the other receptor. In general, all the agonists in high concentration can be a little bit inspecific. So we also did a summatory of all the agonists and all the antagonists, 1 micromolar of each one of them. We didn't see any summatory effect in reducing HIV expression. That was telling us that probably the system was already saturated and the concentration ones were already a little high. So in this case, what we did was we took the 3 agonists that were responding better. We reduced the concentration to 1 micromolar. We treat the H69 for 4 days, and then we did the RT-qPCR, showing here very clearly that the agonists were inducing latency. Again, the A3 agonist, NICA, it was the one that was responding the best. So we took it down to 100 nanomolar. We treat the H69 again for 6 days. Here is not 1 micromolar, it's from 1 micromolar to 100 nanomolar, as you can see in the graph. The Western blot here showing NEF protein expression very clearly. The agonist, the NICA, was reducing latency. It was a saturation happening with 500 nanomolar and 1 micromolar, meaning that probably we don't need that much of the NICA. With lower concentration, we can do better. Also, we realize when you increase the time, probably lower concentration will be also working. And that's what we did. We then went to take the NICA to 25 nanomolar. We quit the treatment for 10 days. One other day, you place the cells in the 75 centimeter square flask, and then you start changing media every other day with the treatment. So 5 treatment, 25 nanomolar. Here, the qPCR also showing the RNA on a splice and 1.8 kV. Very clearly, 25 nanomolar was reducing the HIV expression. But it was this just happening, how this was happening. So it has been proposed the adenosine, the adenosine can be reducing the NF-kappa beta translocation to the nucleus. So we plate the cells in cover glasses, we activated the cell with TNF alpha again, and we start treating the following day with NICA. We treat for 3 days. As you can see here very clearly, the NICA was reducing GFP expression, meaning that it's reducing the HIV. And you can see here the P65 is very much present in the nucleus, in the cells that are expressing GFP. So in the cells that are treating with NICA, you see the morphology is already different, meaning that they are less activated. And also you can see the nuclear area is very much empty compared to the control untreated. That was also confirmed by Western blot here the untreated. This is with the nuclear isolation. This was done by Dr. Feng Ji. He nicely did this experiment for me. You can see here NICA and rigadenosone, both of them were able to reduce P65 translocation to the nucleus. So is this a noise? Is this just happening in the H69, or this is happening also in microglia, primary microglia? So we went there and we took IPS microglia commercially available from Tempo Bioscience, infected with a single round provirus that I use when I create the H69, and then I treat it with a NICA. To my surprise, it was doing nothing. NICA was not affecting the HIV expression in this microglia. So we were concerned, very, very much concerned that probably it was just happening in the H69. So I put this microglia with the astrocytes in co-culture just a couple of days, because whoever has worked with this, if you guys have worked with these IPS microglia, you know that they divide very fast. So you cannot keep it with astrocyte because they overgrow the astrocyte very fast. So then a couple of days, I only saw a little bit of the latency happening. I didn't see anything comparable to what we saw before with the H69. So by that time, our student, and now Dr. Chita Sharon, so she was working doing single cell sequencing, comparing the different IPS microglia. So she nicely put this single cell data in the UMAP. Here you can see the tempo IPS microglia, a human fetal microglia, or microglia isolated from organoid or from assembloid, human assembloid, a microglia isolated from a humanized mice, and microglia that she differentiated from IPS from a scratch. And when she compared those related to the expression of the adenosine receptor, very clearly the major difference was that these tempo microglia were not expressing ADORA3. They have a little more ADORA2B, but they didn't have at all ADORA3. And nicely also they show that the microglia basically they don't express ADORA2A. More likely this is in the brain is more expressed by astrocyte maybe, but more like anything for neurons. So I then start differentiating microglia from IPS. I checked that they were expressing the typical marker, IVA1, P2I12. So I took those microglia, differentiated by myself, and I infected with a single round provirus, GFP expressing, that I used before for the H69. And then 3 days post-infection, I checked that they were expressing the GFP. And then I put it together with the astrocytes. As soon as 24 hours later, we saw that the GFP was getting reduced, meaning that the HIV expression was getting reduced. But I think something very interesting came from this experiment, and it was, I was using cell trace violet, that the monoculture microglia were very different from the microglia that were in the astrocycle culture or in the tree culture. As you can see here, very clearly this experiment was showing that astrocyte and astrocyte neuron are able to modulate the homeostasis, change the phenotype from more rounded, more activated to more processes, probably bigger microglia that they have really, really more processes. So that was very interesting to us. We then stopped comparing monoculture to co-culture. We started differentiating microglia with astrocytes. As you can see here, probably astrocytes are able to modulate the maturation of the microglia just as soon as a 9 day after the co-culture or 9 day after differentiation. You can see the microglia already, they have more processes, they are bigger. Here at day 14, you can see better the microglia in the co-culture with astrocytes are really nice. Then day 17, the microglia are really, really different. So they probably are very different. We haven't done any single cell analysis, single cell sequencing analysis, or bulk RNA, but we believe they are expressing different protein, and probably the maturation is just different. So what I did at this point was, okay, let me take my microglia. I will not be doing co-culture to compare monoculture and co-culture, but then I know the agonist for adenosine receptor 3 is inducing latency before, so let me see what it's doing. Exactly, 3 day post-infection, I treat the cell on day 4 and day 5 with 1 micromolar of NICA, and very clearly this was only two, I have N2 just, but very clearly the adenosine receptor agonist was able to silence HIV in those cells. When I did co-culture also that I treated with NICA, very clearly again, only N2. I need to repeat this experiment again, but it's very clearly that the astrocytes are able to induce latency, probably, I mean, the NICA is able to induce latency in the co-culture. This also in the ICOR, cortical neuron differentiated from IPS, with a microglia, that I put together day 3 post-infection, and I treated on day 4 and day 5. Then we did RT-qPCR at day 6. You can clearly see that they also, NICA is able to induce latency in this condition. I think the more potent situation is in the tree culture. ICOR with astrocytes and microglia carrying the HIV. When they were treated with NICA, 1 micromolar, so the silencing, it was very clear. So the following that I did, it was just differentiate microglia or in monoculture, in co-culture with astrocytes, and then I start using the HIV wild type macrophage tropic ADA virus. I infected the culture with 13 nanogram of HIV, and then I started it 24 hours later. I treated it on day 3 and day 5 with a 1 micromolar of NICA. So very clearly, here this N3 already, you can see how NICA can reduce HIV replication in the microglia, not the antagonist. Also in the co-culture astrocyte microglia, you also can see very clearly that NICA can be reducing the HIV transcription, no like that, the antagonist. Just for you to see the co-culture, for example, here you can see untreated, the cell look after infection and, before collecting the RNA and DNA. You can see that the microglia here in red and green. This is a T tomato IPS line that we use to differentiate microglia, so that way we can follow the microglia or the hematopoietic cells in the co-culture. You can see these big cells, when you do the staining, these are syncytium happening. You can see that the microglia are more activated, they have less process. No, like that when you have the NICA one micromolar treatment, they still have more processes, you have more microglia, and you have less incision. That is also happening a little more with the antagonist. So at this point, I wanted to test the next model that we have been working with. In this case, I'm going to be talking now the organoids or the assembloids when they have microglia. So usually, this is a protocol that have been established in our laboratory by Dr. Cheatham. I have done some modification. I take myeloid precursors that I have been expanding by for 5 days. I take 3,000 of those cells and I put together with 7,000 of neural progenitor. Here we use the T tomato microglia, so you can follow the microglia in the organoids at day 5 or in the assembloids. At day 5, you already see that they are around half of the millimeter. At day 25, they are double the size. And then at this time, yes, you already see that the microglia is not very much in the center of the organoids, probably because the conditions in the center are not very nice for the microglia. So they tend to go a little more to the periphery, but still they are a little bit deep in the organoid, but not that deep. So in any case, at that time, we did some staining, or in this case, it was Dr. Chita that she had done some stain. You can see MAP2, you can see IVA1 and a nice distribution of the microglia in the organoids. Then she also isolated the microglia from those organoids. She has been showing that the amount of microglia in those organoids is very similar to whatever proportion we have in the brain. So I took those assembloids. I then infected with 15 nanoround of HIV, macrophage tropic, ADA. I did the time course of the infection. We saw that probably the peak of the infection is between day 6 and day 8. So we took day 6 as a reference. Uh, we again infected the organoids in a new experiment, and then we treat 1, 3, and 5 day post-infection with NICA 1 micromolar. We then isolate RNA, DNA, and we saw that this is only done once. I had 2 replic. In each replic, I have 6 organoids, very clearly, and it's very promising that the organoids or the NICA is able to induce a very potently it's a reduction of HIV or expression in the microglia or in the organoids. Um, there is a lot of variability, so I need to work in this experiment to be able to achieve the right end and to do the statistical analysis. Um, so at this point, we already start working with the receptor. We believe that ADORA3 is probably the most important receptor in this. In humans, we have 3 isoforms that are also known by isoform A, B, and C. They are just basically produced by alternative splicing. Each isoform is different size. The AB, they differ in the C terminal. A and C, they are differing in the N-terminal region. So Dr. Kim, nicely, he created a virus for me, lentivirus. He took the coding sequences. He cloned those sequences in this vector. It is a blasticine resistance, so he created the lentivirus. I took that lentivirus and I infected the H69. Sadly, I cannot do this in the IPS microglia, so I did an H69. I selected, and after selection, I treated with TNF. You can see here, just 24 hours after activation, the isoform B and the isoform C were already showing less activation than the mock. So at day 6, work very clearly, the cells overexpressing isoform B, they have 20% less HIV expression than the mock. And just for you to see here, the time lapse of this experiment, very clearly the isoform B overexpressing cells, they have less GSP expression compared to the MOG and the isoform A. So more likely, probably the isoform B can have a role, or isoform C can have a role in silencing HIV in the system. So with this, I'm gonna stop my presentation. I just wanna show you that my future direction are to continue working, determine how adenosine can be silencing HIV, more specifically in the astrocytes microglia crosstalk. I also want to confirm which receptor is involved. We believe that the A3, and also we need to know if there is any difference between the isoforms involved. I also want to start exploring if the presence of one or other ADORA isoform could be correlated with the progression of the infection in the HIV in microglia, but also if they can have a role in the progression of the HIV in the HIV-associated narcotic disorder. So I want to thank, and the conclusion, I'm sorry, I was too fast. The conclusion is that, the data from this culture are suggesting a regulatory homeostatic mechanism that could be explored to reduce inflammation and HIV expression in the brain. One of those mechanisms could be related to adenosine. And I think it's very important that our results support the importance of studying the crosstalk among different brain cells types to identify possible therapeutic targets. So I want to thank Dr. Khan. I also want to thank the lab members that were involved with me in this project, the past member, especially David Alvarez Carbonate, my student, the student that we have in the lab, Kim. She was doing some of the experiment, and also Dr. Wilber Romero from Vanderbilt University, the NIH, the National Institute of Mental Health, more than anything, the National Institute of Drug Abuse, that we have some grant, and the money basically is coming from them in all this experiment. So this is all. I am ready for the questions.
Thank you so much, Joelvis. Well, I think
Thank
you
you.
made the case for your point in your summary slide about these complexity and the interactions between the different cells in the CNS. So, Dr. Geiger, did you had a comment in the chat? You can go ahead and ask your question or engage in the discussion here.
Okay, I'll start it off. Very nice presentation.
Thank you.
But I do have concerns over the micromolar concentrations that you used of the adenosine receptor agonists, and specifically NECA is clearly a non-specific, non-selective, certainly at micromolar concentrations for 3 of the 4 adenosine receptors, the A1, A2A, and A3.
Mhm.
So all these agonists have, KIs, KDs in the low micromolar range, nanomolar range. So when you start putting them in at tens of micromolar, they have no selectivity whatsoever.
Yeah, so I think we have talked about that, or you have been trying to make this question in other presentation that I had before. We talk about a little bit about this. When I present this work in Montreal, I think we talk about in the ISMB, and I say yes, there is high concentration and there is a specificity. But however, I show today this data is new for you that I work with NICA in the 25 nanomolar, in the 100 nanomolar. So at that concentration, NICA is very specific to A3 receptor. We are also working with the cell line. Usually the cell line are a little bit different, somehow in the brain, you can have magnification of the signal. You can also have depression of the signal. We are working with concentration now that are more similar to wherever we believe is right. So when you are working with IPS microglia and you have infection with a wild-type virus or even with a single round virus, and you put together a microglia with astrocytes, so those microglia are already mature. They have a limited life. So you cannot be working with a low concentration and longer term because you will not have results. The microglia will be dying at the end of the day, So, and also when you put the virus there and you start working with the wild-type virus and the virus start replicating, you will also be running with microglia. Actually, this is data that I didn't show here, but if you keep this experiment for 9 days, so then the microglia from the NICA will have more virus than the other. And the reason is just because the other cells were already saturated, the infection was already gone, and then the NICA, because it was keeping the cell healthier, it will be then showing more virus. For that reason also we took 6 days as the limit time to show this data. Again, so we understand that 1 micromolar. However, as I said, these IPA from Tempo are showing that they don't have the A3 receptor. And when I use high concentration, even 25 micromolar, it was not silencing HIV. So I understand the concern related to the inspecificity of the agonist, but I think we have proved that probably is this through the ADORA 3 receptor, and we also use 25 nanomolar as a very low concentration that they can be working through the ADORA 3 receptor.
Thank you.
Yeah.
And potentially just following up on that, another way, or is it possible to specifically knock out the receptors?
Yeah, so this is something very interesting. So they are not really good antibody for the ADORA 3. There was this antibody from Santa Cruz that people used years ago. They were saying that the receptor was a 48 kilodalton, which is really no. I saw people showing that there was modulation. There were modulation for that receptor. I tried to get the antibody. It's not there anymore. No, they don't have it, and I cannot find it. The receptor antibody that I took for ADORA 3 is just detecting the ADORA, the isoform A, the 38 kilodalton very nicely. Where we don't see any modulation, we try to do a knockdown. In the A69, we didn't see anything happening. So that's why we start, okay, let's go and start to overexpressing the receptor when we have this overexpression very clearly with phenotype and with a flap. Then we can start work with a knockdown to be sure that we are really knocking down, taking back the phenotype, and then probably working with some interference RNA in the iPS microglia. Yeah, we were also already talking about knocking down the gene in the iPS, but it's something very difficult. The iPS are very sensitive. They get self-differentiated very easy with any modification that you do. It was very difficult to get this 3D tomato iPS just because of that, Yeah, we are working on that, and hopefully we will be able to achieve what we want later on. Yes. You are on mute.
Sorry, Dr. Geiger just put in, should people with living with HIV not consume caffeine?
Oh, well, the caffeine, is not really doing anything over the pathway, So basically, it's linked to the receptor, but it's not really affecting the pathway in the way that is inducing activation or inducing inhibition of the adenosine pathway. So yeah. Other way, we are all good because we take a lot of caffeine, I think tea or whatever, So yeah. In any case, also,
Go ahead.
I want to say something that I thought to say in the presentation and I didn't say. This nice paper where they show all the pathways and all the targets. These people are from Pittsburgh, and I wanted to recognize that they have been working a lot with that. I was reading their work, and I think it's very interesting. Yeah, that I wanted to say that. Yeah.
Good. I was curious about the the iPSC derived cells, right, are from different individuals, right? So, is there a role of these line derived phenotypes? But I guess in your later studies, you did use two different lines.
So I had used different line for the microglia. They are different when you are differentiating them. Let's say the iPS, the T tomato iPS line, the clay line. So those are coming from fibroblasts. They are a little bit different. Also, astrocytes coming from fibroblasts, they are a little bit different than astrocytes that have worked coming from the PBN2 line that they are coming from the blood. So they behave different, they differentiate different. But always the astrocyte different. I use 3 or 4 different astrocytes from different iPS line that I could culture with microglia. They were always inducing latency. Also, Nika was always inducing silencing HIV also in the different microglia. I think the issue here is to get a nice iPS microglia from different line. These have been the more difficult for me.
Okay, great. We have some questions, Dr.
Yeah.
Eugene, and then Dr. Gaskill. Go ahead, please.
Hi, Joelvis, and good
How
to
you
see
doing?
you, Amanda. Good to see you, Gaskill. Then, did you analyze in any ways expression of CD73 This is question one. And the second one, how you select the times to treat with NICA? I see that you treat after 1 day, 2 days, 3 days. Is any rationale to do that? Is any way that adenosine, for example, is blocking the entry of the virus or something like that, and that's why you have that difference instead of replication?
So, well, there are a few things here. The first one related to the timing of the NICA. So I first was doing, high concentration of the agonist in a short time. When I started reducing the concentration, I'm sorry, I saw that there was saturation in this. Let me see if I can show you here. Exactly. So in this experiment here, you can see I changed from 10 and 5 micromolar, even I use more micromolar, to 1 micromolar. 1 micromolar was also inducing latency. And then I changed NICA, that was the best one. I changed it to 100 molar. And then, here there is saturation of the system already. I increased the time. Sometimes the increasing of the time was telling me that you can keep the cells longer with the treatment and they will respond better. That's something that you see because sometimes accidentally you don't have the cell were not growing as good as you were thinking for any reason. Some of these experiments sometimes are done by students that they are learning, but you can see that the cell are taking longer to grow, and then that was taking me to longer to do the experiment, and I realized that longer treatment with lower concentration was as good as higher concentration in a short time. You understand? So that's why.
But the point is like yes, I get the concentration but why you select 1 day and 2 days, 3 days?
I didn't. I mean, so you can start with 1 day and treat them only 1 day, but I never did a 1 day treatment. I did always 3 days. I try to do at least 2 treatments. So you,
Okay.
2 or 3 treatments, so you have the microglia that is differentiated at day 28, around day 28. You infected with the single round virus. 3 more days is already 31 days. Whoever worked with microglia already know that microglia differentiated from IPAs already all at that time. So if you treat the microglia for 3 more days, they are already 34 to 35 days. And that's what I try to do. Don't keep longer the microglia so that I don't get into kind of dying my cells and misleading the experiment.
Okay, and the CD7 and the CD73 expression.
Oh, the CD73 expression.
Yeah.
So in the same, the microglia are expressing a lot of CD39. They are not expressing almost CD73. They express very low level. This graph here, the new map graph, I think it didn't was important because in any case, the CD73, it have been very much reported that is inducible, and it's inducible for in different conditions like low oxygen or with TGF-beta and in other factors. So I didn't present it because it's not really. I thought it was not the most important, but the most important was just the ADORA3, and this was the major difference. But none of them are expressing basically any CD73, something very similar to this one here, even lower, but they express a lot of CD39. Okay. That is the other enzyme involved in the catabolizing the ATP and the ADP.
Thank you so much.
That was all your question, no? Yes.
Go ahead, because we're running out of time. Go ahead, Peter, please.
Hey, Elvis, thanks for sharing your data. So I just had 2 quick technical questions. You've used a couple of different types of iPSC microglia, I think. Were they male or female lines? Because one of the things I've been discovering when I talk to different companies is that female lines are much more complicated to work with than male lines because of the expression of 2X chromosomes. And I'm just wondering if you've seen that in your study or you're just using a male line of microglia.
I use different line, but I cannot say from the top of my head. I know there is a male and there are like 2 females, but I cannot say from the top of my head which one it is. As I was saying before, my difficulty with this is in the differentiation because some microglia, when they are differentiated from some IPS, they get more activated easier than other, But no, I didn't find difference in the responsiveness. So, when they are together with astrocyte and the neurons, they basically all behave the same. When they are in the monoculture, it's more difficult.
That is really interesting. I was going to so one of the things that we have heard from a number of different companies is that when you have female iPSC lines, because of the variable expression of X chromosomes over passage, that they are much less consistent in monoculture, but you're not seeing that in co-culture or triculture.
No, in co-culture I don't see really a bit different, but in monoculture I can see. Actually, sometimes I am preparing my microglia for a monoculture. And by day 10, day 12 is when I am adding the microglia to the neuron or to the astrocyte. I decide, no, this culture will not be good for monoculture because the microglia are not looking very happy. And when you add the co-culture, then quickly they start, like going smoother and they start being nicer.
The other question really quick was about your assembloids. I saw you only took the infection out to day 10. Did you take it out longer at one point and then just decided day 10 it looks the same?
No.
You're back there?
I think you cannot take it longer because as you can see here, the peak of the infection is around this time. So it meaning here is that the microglia are already dying, because of the infection. So the cells are dying after the infection. So we know that we still have microglia at this day 30 something. But
do you think they're dying or that they're just crawling out of the assembloid? Because they move out from the middle and they get to the edge. Maybe they just
because of the infection, so they can go out also, So if you take the assembloid longer time, you will have less microglia, that's for sure.
Yes.
That's why, this is the different in this mode. Many people have different model for the assembloid.
Absolutely.
In our case, we are trying to improve it, but we see that the microglia, we didn't check how long the microglia will be there, but we know that if there is any microglia, it's not a big amount to be doing this experiment. So that's why we're taking only to those days. We talk about that,
Yeah, absolutely.
Yeah.
No, I think it's interesting. We see a similar thing that we see an early peak in infection followed by a decline over time. Ours lasts a little bit longer, I think out to about 20 to 25 days. Before we start to see the decline, but it's definitely something we have seen them move to the outside of the
How much virus do you use?
How much virus we use? Well, we've done it with 530 and 100 nanograms of virus. So you can use much less, but you still see about the same amount. So we think we're saturating our system, right? So if you tried less HIV and you still saw the same amount, it would tell you you're saturating your system. You might be able to use less and cause less toxicity in your microglia.
Yeah, also you can use less and it can go longer. But so usually with this we thought Cheetah have been the one that have been standardizing this the most, and we have been following, a little bit her steps.
Absolutely.
Okay, great discussion. We must continue this, but I'd like to get to Dr. Goodkin. Would you like to unmute? Dr. Goodkin, you still there? Yes. They have a question in the chat. Meanwhile, Elvis, you can read. This is more of a clinical relevance question since extracellular NAD can be degraded into adenosine, and it may produce its effects through this degradation, and it can also increase the pool of microglia. What are your thoughts about?
I'm sorry. I need to hear the question again.
Okay, it is in the chat. It's kind of long. Dr. Goodkin, are you on?
Yes, I'm on.
Okay, go ahead.
So I was wondering, since extracellular NAD+ can be degraded into adenosine and it may produce effects through the degradation and can also increase the microglial pool, what are your thoughts about NMN as a nutritional supplement that's converted into NAD+, improving mitochondrial function and neuronal function in brain and presence of HIV, as well as having senolytic effects, which is important in people with HIV since 50% of people with HIV in the US are over 50 years of age.
So to be honest, it's not that I don't know. I don't like to be speculating about something that I haven't read at all. But in any case, these nutritional factors somehow helping to increase the level of adenosine. It may be good. In any case, we are using agonists. It's not the same. And I can tell you that when you put adenosine in the system and you increase the concentration, what will happen is this adenosine will go through the transporter to inside the cells, or it's gonna go to inosine by adenosine deamylase. So they try to keep the concentration of the adenosine in the right level, So it's just a normal way for the system to work. The same way that when you increase the concentration of ATP, at least in kind of acute system, the ATP can be catabolized to adenosine to keep the balance and to keep the homeostasis. So yeah, it may be good to have this nutritional supplement, but I cannot say for sure that this is really increasing the concentration of the adenosine in the brain. In the periphery probably it's easier, but in any case, again, when you have too much adenosine, the adenosine will be catabolized, will be degraded to inosine, or can be transported to inside the cells.
And also, Charles Armstrong suggests consider the phosphorylation of adenosine, phosphorylated forms of adenosine. So this is a discussion to continue. Caroline is on. So thank you for an outstanding presentation and discussion. Thank you all.
Thank you.
Thank you. And for everyone, for next week, December 10th, the speaker is Nicole Perez from NYU, and the topic is disentangling depression in women living with HIV. Thanks, everybody.
Thank you.
Thank you.